So I'm an adjunct professor at Duke in the Department of Medicine. And before I sort of had left academia full time, my son was diagnosed in around 2020 with DYRK1A syndrome. He's 8 now. And I decided at that time, I mean, I was doing research in oncology and hepatology, and I was like, why am I focusing my efforts here when I could be doing it something more personal? So I ended up leaving academia full time, and I work in rare disease drug development in the pharmaceutical industry now, just to be in that space of understanding how you get drugs approved and how to move the needle further. So that's really what my job is and my role is, to help DYRK1A syndrome is really to try to help push that needle forward to get treatments available. And it happens since, my wife, Sarah, she's also on the MSAB. She was actually already in rare disease research before Ethan was even diagnosed. So, I mean, say it was sort of coincidence or, just meant to be. And so, we feel like we're in a pretty good position to help the community, and we have a growing network. And what we've learned through that period of time really is that there's so many researchers and people that we've connected with that are like, we just want to help. And they have. And because of that, we've made considerable progress in a short period of time. I've said it a little bit before, but before he was diagnosed, we were just doing symptom management, and both my wife and I have got in genetics, and we're like, well, I mean, even if we get the diagnosis, like, we're already managing symptoms. Like, what else can we do? I didn't think it was even on our radar that we were going to be able to work on developing therapies for DYRK1A syndrome. And here we are, we're going to be talking a little bit about what we've been working on in that time period. So I know it's sort of long-winded intro, but just wanted to, give some context there. And is it going to want to do it manually too? Okay. All right, so now it's working. So Amy already covered, we've been a growing community. The biggest thing, because we're a growing community, is now that we're able to get more data. And the more that we understand, the more that we're able to do. Rare diseases by their very nature are rare. And so when you're dealing with small sample sizes, from a statistics point of view, it makes it difficult to make determinations, because, clinical trials and a lot of things are run by statistics to determine, if they're efficacious or not. And so by growing our community, by reaching more individuals, this is only going to be to our benefit, not only to make us feel like we're not alone because now we have a growing community, but from a scientific and medical standpoint, this is really what's going to be driving a lot of the innovations moving forward. So as part of the medical and scientific advisory board, we had set up a number of objectives to accomplish. One, first and foremost, was to deepen the understanding of our community. We already know that DYRK1A syndrome looks very different depending on what family meetup you come to. The individuals are all very different, and we're trying to just understand that from a scientific perspective. The second is to establish the drug development toolkit and a pathway to therapeutics. Sorry, I forgot this was still a slide. And I'll talk a little bit about our therapeutic strategy, some of the things that we've been doing in terms of working on developing therapies. The third is to establish a research funding mechanism, so which we did. So Craig's my brother. Craig, he was the first recipient of this grants program, and he presented on his project yesterday. His project is on basically a proof of concept of treating an individual with a very rare mutation in DYRK1A syndrome, who will be most likely the first individual with DYRK1A syndrome treated with antisense therapy. The fourth, which we're still in progress on, is working on defining a center of excellence. A center of excellence is really a place where they have expertise in DYRK1A syndrome. This doesn't exist in the United States. We have some ideas of places and physicians that could accompany a center of excellence, but this is still a work in progress that we have. And then last is educating the community, not just our community, but the broader community on DYRK1A syndrome. We did this using the research roundtable that we had yesterday. We've done these quarterly research roundtables. Admittedly, we haven't done a good job this year for a variety of reasons, but we're hoping to pick that up, and hopefully it'll be hitting your inbox soon about ways that you can join virtually and be able to understand what kind of research is being done with A syndrome. And then lastly, we just had in the Frontiers of Neuroscience, we had a publication on specifically DYRK1A syndrome. So we invited researchers to submit publications. I think we had 5 that were submitted, an editorial by our group that was published, and I'll touch upon that briefly. So what happened in the past year, if you were at the meeting last year, just wanted to provide a quick update. So we funded 2 research studies outside of CREG. So one is another strategy using an RNA therapeutics platform to increase DYRK1A expression. So it's challenging to say, but increasing expression of a gene, of a protein, is probably one of the most challenging things in science today. It is much easier to block, knock down, eliminate, anything like genes like that. From a scientific standpoint, that is much easier to do. And our DYRK1A syndrome is a haploinsufficiency, so you don't make enough DYRK1A. And so increasing that is difficult. There's a completely another layer that's added complexity to that, because DYRK1A is like a Goldilocks gene. So you can't have too much and you can't have too little. So, it makes the science, you need to have a lot more precision with it. So it makes it a little bit more challenging, but, we've funded 2 research studies. So that one is to increase DYRK1A expression. There's also another, we have a collaboration with a researcher at Johns Hopkins by the name of Jeff Collar. He also has another strategy using an RNA therapeutics platform to increase DYRK1A expression using sort of a different type of technology. So we've already funded sort of 2 additional mechanisms to help our community. And both these mechanisms would be considered sort of mutation agnostic. And what that means is that we already know that all our kiddos and patients in our community have different mutations, and this would essentially bypass that. So you wouldn't have to have a specific mutation to have this type of treatment. So that's amazing. An ICD-10 code. I'll go probably into this in a little bit more detail. I'm probably doing too much detail right now, but we'll talk about that in a little bit. We're also doing a disease concept model. So you may have already participated in interviews with a genetic counselor to gain insight into the lived experience of DYRK1A syndrome. So there's information in the literature that says this is what DYRK1A syndrome is, and it's characterized, but it lacks the lived experience. We already know that there's so many impacts to our community that aren't captured in the literature. And so the disease concept model, the purpose of that is really to capture that. And then that data basically will go on to then help our natural history studies, because we look at the disease concept model and say, hey, there's a bunch of things that are impacting our community that we're not collecting, and we need to start collecting this. Like if it's a specific GI measure, specific sleep measure, we can start collecting that if it's not already being collected. And then additionally, in rare disease drug development with the FDA, they always want community input. So that's like the biggest piece that goes into that, because then you have the community voice saying, these are the things that are important to us, not just, what the clinicians are saying, but these are the endpoints that are meaningful to us. So there's a number of important aspects and implications to the disease concept model. We already talked about the editorial that we have, talked a little bit about maintaining participation in the tiger space, finalized with their follow-up of stripes. And then I'll talk a little bit about RareX. And then lastly, the accomplishment that we had in the past year is that we were awarded a grant by the Simons Foundation to support our scientific conference that we had yesterday. So this grant allows us to help fund researchers to come in, talk about DYRK1A, get the discussion. Some of you attended, that yesterday. So it really continues to expand our network, expand interest in the research around DYRK1A syndrome. So Emily Newhouse presented on the Tiger study yesterday, and I think for time I just won't go super into it. But it's been a 10 plus year collaboration at the University of Washington, Genome Sciences Center and the Autism Center. And, there's a number of really interesting data that she presented on yesterday. And that work has been published. But, one of the things I think that was really interesting that came out is that, there was no real single profile in terms of intellectual disability that was found within our community. And I think that just highlights the differences that we see across our kiddos. Some are more severe than others, but there really isn't any single profile. And I thought that was really interesting. But Emily did a great job presenting on that. And we can also invite her to the research roundtable and talk a little bit more about the Tiger study if you're interested in learning more. I'm going to put a plug in for RareX, so you can actually access the data collection program through the DYRK1A website, and I would encourage you to do so. I think there's like a research tab. If you click on it, you can go now, and then you can get started on the RareX platform. This is my wife, and that's Ethan right there. He's a lot younger, but I would encourage you to go into this platform because what you'll do when you sign up is you'll upload your genetic report, and if you receive the genetic report for your diagnosis, which probably most of us did, and so once you get that, you can upload it. It'll be validated by a genetics counselor who's part of RareX, and then you would go in and do a bunch of these surveys. And I know that it's like cumbersome and it's time consuming, but really like these surveys are the best snapshots that we have right now for a natural history study. And these natural history studies are absolutely essential to the drug development process because, clinicians want to know, like, what is it impacting? Like when you design clinical trials, what endpoints do we want to look at? Because if you're not selecting the right endpoints, if you're not selecting the right things to measure, trials can fail, drugs don't come to market. So this is critical just to understand, our community and just to like bring up, some snapshots. There's a sleep habits questionnaire survey that was in there. And, over 60% of respondents had a score of 41 and above, which is actually a characteristic of having a diagnostic sleep disturbance. So, we wouldn't have this information if, folks didn't go in and, fill out the surveys, and from this information, you can gather what type of systems are impacting. If we know the types of systems that are impacted, then we're going to want to, dive into that further and get more information so that, we can probably, get assessments for that. Again, this is information on like feeding difficulties, constipation, autism spectrum, coordination issues, seizures. Again, capturing this data may seem like a no-brainer. Like, yeah, we know what's going on in our community, but like this is the type of data that we have to show in order to help develop, the information needed to design clinical trials. And uh, Yael Shiloh-Malowski was here yesterday, so she's a neurologist from UNC, and she talked about the need to develop clinical care guidelines. We have no clinical care guidelines for DYRK1A syndrome. Imagine going to a physician and being able to have clinical care guidelines where okay, we actually need MRIs done every 6 years, for instance, or every 10 years, or we need eye assessments done at least every other year, something like that. Like we don't have any clinical care guidelines, and this is the type of stuff that would feed into that, because what systems are impacted and what you need to assess. I'm going to go quickly into a proteomic study. So a couple years ago, we had a family meetup where we had blood collected from kids with DYRK1A and their families. And one thing that we were able to do, because we're part of this larger consortium of neurodevelopmental disorders called Combined Brain. So what the power of Combined Brain right now is that we're saying, okay, well, we've got this small community that has DYRK1A syndrome. It's a neurodevelopmental disorder. What if we take that neurodevelopmental disorder and we combine it with like 50 others? Now you have a massive data set of neurodevelopmental disorders, because there's a lot of overlap. I mean, there's a lot of symptoms that overlap and pathways that overlap. And so now you have more statistical power to start asking some questions. And so we did that. So we had 19 individuals that had DYRK1A syndrome and 19 controls. There's Vineland scores that indicate sort of moderate developmental impairment. Vineland is part of that RARE-X survey. And what we ended up finding in this sort of data set is that there was some plasma markers in the blood that were significantly higher in DYRK1A patients that were also indicated in other neurodevelopmental disorders, and that when we kind of clustered them together, they mapped with other sort of related epilepsies that impact like synapses and channelopathies. I won't go into like the specifics of all the data that was found, but the take-home message is that because the community provided samples, because we were able to combine this with a larger data set, we're able to get some learnings about commonalities with other developmental disorders. And so then you can ask more specific questions about genes that are affected, and are these genes important? Should we be studying these genes further? So it's just a really good testament to sort of the collaboration that happens. Even though we're a small community, we can be a part of something much larger and gain actually insights, even though we don't have, large numbers of individuals. So I'll just go really quick into the disease concept model. So this disease concept model I mentioned is really important for clinical trial development, and it really gets the lived experience of DYRK1A syndrome. So the nuts and bolts of it are based upon interviews with caregivers and their families. It's also combined with interviews with clinicians as well. And what after you can, so what you do is you compare the sort of lived experience with what's in the literature. And this is just a massive little slide for some reason. But what this shows is that over 50% of what was reported in the interviews were completely new concepts that weren't described in the literature. So this is really important because now we're getting like a whole different number of concepts that are new, that are important to our community, that aren't described elsewhere. And so again, combining this with what's reported in literature and the clinicians getting on board can help develop clinical care guidelines. This can help develop pathways to developing clinical trial readiness. So, to everybody that's participated in this, really appreciate that because it's so critical to moving forward. I'll touch upon the ICD-10 code. Does anybody in the room know what an ICD-10 code is? All right, so we got a couple of hands. So an ICD-10 code basically is a coding system that's used for like diagnostic purposes. It's used for billing. It's used for a number of different pieces. But DYRK1A syndrome doesn't have an ICD-10 code, and in fact, there's thousands of rare diseases that don't have ICD-10 codes. But it is absolutely essential to have an ICD-10 code, especially if you want to do clinical trials, especially if you want to understand how individuals are treated over time. You can just pull an ICD-10 code and say, okay, this is an individual that's diagnosed with DYRK1A syndrome. This is how they're treated. Or you can look at all other aspects related to their care. So we've been working on developing an ICD-10 code. I won't get into like the logistics of government bureaucracy, but it's been tough for the past 2 years to try to move this because there's very few people in the government that are kind of working on this. But again, through our collaboration with Combined Brain, we've been working on this Mondo system. It's basically just a system that tracks the use of codes through other groups, and it helps assign codes essentially more quickly. And so we've been working with that. So we have fingers crossed right now that we're going to get an ICD-10 code for DYRK1A syndrome this year. So hang tight. But I think this is something we've been working towards. It's going to be very, very critical to our community moving forward. So hopefully I'll have a positive update soon. I touched upon this was a grant. It actually wasn't a grant. We just straight up funded it because it was another collaboration here with Combined Brain on. It's called an mRNA booster, but the purpose is again to increase DYRK1A expression. So it's another strategy to help increase DYRK1A. It's mutation agnostic. Any China has a DYRK1A mutation outside of gain of function mutations would be able to potentially benefit from this. I'm just going to say this is all very preclinical data. They've used it in other neurodevelopmental disorders like CTNMB1, SYNGAP. This is all preclinical. This is like mouse stuff. This is in lab. This is like not close to getting ready for prime time. Tested DYRK1A. So this is very early on, but it's very promising technology, and we're hoping to get some proof of concept working DYRK1A very, very soon. But these are honestly some of the best technology platforms available right now to do this type of strategy. I talked about the Frontiers of Neuroscience paper. It's had over 22,000 total views, almost 5,000 downloads. The content is not important to tell you about right now, but what's important to tell you from these numbers is essentially this is us branching out our network to the surrounding community, getting people interested in knowing what DYRK1A syndrome is, and trying to essentially get researchers to get interested in and trying to study it. So that was the whole point of doing this, this scientific articles to try to get, some bigger interest in DYRK1A research, because the vast majority of research in DYRK1A syndrome, or in DYRK1A specifically, is in Down syndrome. So I'm sure many of you are aware that, DYRK1A gene is on chromosome 21. In Down syndrome, it's basically 3 copies of DYRK1A. So there's tons of data on Down syndrome DYRK1A. There's very little data in DYRK1A syndrome as it relates to DYRK1A. In fact, most researchers try to not let DYRK1A because that's what people want to do in Down syndrome. So like I said, most of the research has been done on that other end of the seesaw where you're trying to like turn it down, and to try to increase DYRK1A is much more complex and much more rare, because as DYRK1A stands, it's involved in lots of things. Pretty much anything in the body DYRK1A is always involved in, and in most of those situations, it's overexpressed, and that's considered a bad thing. So they always want to knock it down. There's like very few research to try to increase it. So that's part of the hill that we've been climbing up for the past several years, is not only to try to work on the strategy to increase it, but to increase understanding of how to do that. So we have a competitive grants program. Craig is over here. So Craig was the first recipient. He's been working on kind of one project, proof of concept. We have a number of high priority areas that we focused on. So we don't have just like an open-ended grant like, hey, if you're interested in DYRK1A syndrome, go ahead and apply. We have specific topics and areas that we want researchers to focus on when they submit their letters of intent. So we made excellent progress to date. It's happened in a short period of time, so we're pretty happy with the way things go, but we're definitely not settled on it. We're trying to push it all as far as we can. And part of that is relying on the community, your involvement in research, your involvement in natural history study. We won't be able to make these strides for you. So I would encourage you to try to get involved as much as you can, as much as you want to, because we need everybody's help to move this forward. That's all. I can't remember.
Right.
Am I right on time?
You were right on time. Yep.
Okay. Cool. Does anybody have any questions or is there anything?
If and when the center excellence goes into being, would it be advantageous? I think I know the answer to this, but when I ask it, do you have our kids, people with the syndrome, actually physically attend the center of excellence to build that syndrome, how to treat it?
Yeah, I think that's a great question. So the idea is to set up multiple centers of excellence. So we would obviously start with one in the United States, but the idea is to grow that to multiple centers, and a lot of that is related to diagnostic rates. Obviously we've seen our community grow. And so if you're in an area or if you're in a state where there's a lot of individuals with DYRK1A syndrome, then I think having a place where there's a clinician who's more interested in DYRK1A, seeing a DYRK1A patient, and getting more individuals to go to that center, those are like ways to help kind of establish that, because then you're seeing more patients. You have a center of excellence where they also have a multi-disciplinary team, and that's part of it too. Like we know that there's so many systems that impact our kiddos that you want a place that has specialists like in all those areas on site that is seen on that and know how to treat. So I think that's part of it too. But it wouldn't necessarily be like we only have one. We want everybody to go to that one. We want to just set up that one first, and then establish more robust treat that can hopefully sort of mirror.
As far as the next step for people who haven't done anything yet, but it'd be the rare MS, the one people to go to, that's the main repository.
Yeah, so the rare X platform isn't perfect, and I know that there's probably some fatigue, in terms of like filling out surveys. I mean, pretty much every time you go to the doctor, you're going to fill out surveys. But as it stands right now, that's probably our best natural history study sort of database. I think there's others that we're trying to look into to make it like easier and so like phone more for our data. But the RareX platform is like the best thing we have right now because it's going to have a validated clinical reports. You're going to upload your mutation status. I mean that and the surveys that are in there are validated surveys, and they're like the greatest surveys that we have for our community to understand our community, and that's where we have probably the most data. So I would say that if you're able to RareX platform for sure for now, and if we can change it to something that definitely actively looking into those things, but for now that's probably the best system that we have. And what I'd like though in the future is that we can have maybe like a clinical coordinator or somebody who can call nowadays to follow up like another report. So that you do it once and then be done, because one of the things that we also have is longitudinal data, and what that means is that so if I years for some of these surveys that I pulled out, and every 5 years maybe surveys, so they see how it changed over time. Because even from a biological standpoint, looking almost you see that genes are changing over time from pediatrics, to adolescence and adulthood. So some of them surveyed and they all can change over time, and we don't have good data or much data involved in older individuals. Right? So, because the diagnosis of those aren't really there, so we don't have a lot of like that or that. So we can follow our community over time and then help us understand sort of the trajectory of sort of my syndrome. And even if it's different for everybody, see how it's changing over time. I think that's going to help us, make better assessments moving forward. Well, I really, it's a pleasure to speak with you all this morning. Thank you for being here. Thank you for being involved in research and for staying in touch.
And introduce yourself. Hi, I'm here, so I'm here. I